This is the molecule of DNA. All of the fundamental instructions for life on Earth, encoded in 4 unique bases, are contained in these tightly coiled molecules. But how are these instructions read, and how can scientists modify them to change the way that organisms work? Let's take a closer look at DNA's contents for the answer. Your DNA is divided into small segments called genes, each of which are transcribed into messenger RNA and translated into proteins, the functional molecules that actually perform tasks for the cell, which include regulating transcription for other genes. When a gene is read or transcribed into RNA, proteins called transcription factors bind to the promoter region of DNA just before the gene, forming a transcription complex. Then the gene is unzipped from the middle by the transcription complex as it moves along the DNA strands, and the protein RNA polymerase in the complex builds a strand of RNA using one of the DNA strands as a template. When the transcription complex reaches the terminator DNA sequence at the end of the gene, it can stop reading and release its RNA product, which is then made into a protein by other parts of the cell. Since DNA is common to all organisms, genes from animals, like jellyfish, can be transcribed by bacteria or plant cells as long as the cells recognize the promoters and terminators around these genes. Synthetic biology works based on this principle. Synthetic biologists insert artificially designed genes, promoters, and terminators into host cells, which could be any species from bacteria to an animal cell, in effect engineering the organisms to produce useful proteins or perform services. Synthetic biologists are working on standardizing promoters and terminators across many different organisms, which would make it easier to use the same gene sequence in different applications. Scientists are also building simple logic gates using DNA and protein products. For example, they can use one gene for protein A and another for protein B to regulate a third gene for protein C that is transcribed only if both proteins A and B are present. This is equivalent to an AND gate, where both inputs must be on for the output to be on. Scientists can also build OR gates, where the third gene is transcribed even if only one of the earlier proteins was produced. Using simple gates like and OR, the logical building blocks for complicated computations, is a huge step towards being able to program cells like computers, which would have countless applications in fields like medicine, materials science, and environmental science. New discoveries and technology in the field of synthetic biology would eventually give us the power to design complex multicellular organisms. And now that you have a better understanding of synthetic biology, you can help make those advances and help bring synthetic biology to the world.